Okay, good morning, good afternoon, good evening, everybody, and welcome to the first part of chapter 15 lecture where we are going to be talking about the special senses. And today we are going to focus on two special senses. We shall look at the sense of smell and the sense of taste. Then in class, we shall continue with the rest of them, the sense of vision and the sense of hearing. So, welcome. So first, remember that we have two types of senses in the body. Excuse me, the general senses and the special senses. And we already talked about the general senses in the previous lecture, where we identified them to be those senses that are found all over the body. So special senses are senses where the receptors are localized within a specific region of the body. They are not all over the body. Specifically, they are found within the head, the region of the head. And they include olfaction, which is the sense of smell, sense of taste, sense of vision or light, and hearing and balance. We normally study hearing and balance together because the structures that perceive hearing and the structures that perceive balance are found in the same organ. They are both found in the ears. So those are the special senses, olfaction, taste, visual, and hearing. Like I said earlier, we are going to focus mainly on the first two today. Starting with olfaction, the sense of smell, this is sense receptors that have been specialized to perceive presence of chemicals in the air that we breathe in through the nose, and are able to detect the presence of those chemicals and send information to the brain. And the brain then converts or interprets what that information means, and then we say we are smelling something specifically. In other words, these particular receptors are chemoreceptors, and I'll ask you to look at these two pictures. Well, it's a single figure actually in your textbook, so that you see the location of the receptors that perceive smell in the nose. The first picture there shows a sagittal section of the front part of the face, where you can see the nose just above the mouth, and you see those structures called the concha, so that when the air is going in to your mouth, rather into the nose, it has to pass through the epithelium lining the top part of the nose, and that is where you have the receptors localized. And the picture at the bottom there shows the actual specialized cells that are able to perceive presence of chemicals in the air you are breathing in, and convert them to action potentials that are then transmitted by those neurons, sensory neurons, taken to the brain for interpretation. They are generally accepted that it's been accepted generally that there are 7 primary odors or 7 primary smells, but in general, we are able to perceive several thousands of different smells or different odors, about 4,000 different types of odors. And the structures that perceive that information is found in the nose is a modified epithelium that we call the olfactory epithelium. So check out that figure and be able to identify each of those different structures in the book. And this figure shows in a little bit more detail how the presence of a chemical in the air is able to initiate an action potential, how it's able to trigger an action potential. And so a chemical that's in the air, we are breathing in, binds to a receptor in the binds to a receptor in the nose, in the epithelium, and the binding of that chemical called the odorant binding to its receptor triggers a series of communications. I don't know if we covered this before. Ways of communication between the receptor and voltage. These are ligand-gated sodium receptors or ligand-gated sodium channels. So when the odorant binds to its corresponding receptor, the receptor somehow communicates to the sodium channel to say that there is a chemical that is bound here, and so you need to open. The communication system is kind of complex. We won't go into details about it, but I'll just mention here that's involving a series of proteins that are found on the membrane called the G protein. And when the odorant binds to the receptor, that triggers the G protein to become activated. And when it gets activated, the G protein that is consisting of alpha, beta, and gamma subunits are split apart into two, where the alpha separates from the beta gamma subunit, and the gamma goes to bind to another membrane protein called adenylyl cyclase. That protein is adenylyl cyclase is an enzyme, and the enzyme, what it does is to convert ATP to cyclic adenosine monophosphate, and the adenosine monophosphate is a communication chemical within the cytoplasm, and the cyclic AMP then goes to bind to the sodium receptor and causes it to open. And the sodium channel, not a receptor, is a channel. When it opens, then sodium rushes into the cell. And when sodium rushes into the cell, that is the beginning of depolarization. And now that depolarization or that action potential is then transmitted all the way to the brain, and where the brain converts, interprets that information as smell. And it will interpret it as a specific smell. And follow the step-by-step in this picture, starting from number 1, 2, 3, until you understand how the presence of a chemical in the air you breathe in ends up causing an action potential. That has been summarized there very nicely. In this picture, we have seen it before. It's showing you the components of the olfactory epithelium, showing you the specialized cells called olfactory neurons that have processes going into the nose in the form of a cilia. That's where they will perceive the chemicals in the air that we breathe in. And each of those olfactory neurons is connected directly to the olfactory bulb, where they transfer their information to the olfactory tract that will take that information all the way to the brain for interpretation. So again, check out this figure in your book and be able to identify the different components of the olfactory neuronal pathways. Be able to identify the olfactory epithelium, connective tissue underneath, the bone, of course, the upper palate, and the olfactory tract, and so forth. That's basically all I needed to tell you about the sense of smell. First, that the sense organs of smell are found in the nose, that they are chemoreceptors. They detect chemicals present in the air we breathe in, and they send that information to the brain for interpretation, and that we can detect several thousand odors, about 4,000 of them, different types of smells. We are able to detect that. And I forgot to mention that this epithelium gets replaced very, very easily. So like every 10 days, the epithelium for smell is always being replaced. Let's switch on to the second one, which is taste. And the sense of taste is very similar to the sense of smell, and in fact, they are interconnected at some point. Say that if you have ever had flu or a cold where your nose is stuffed, then you might have realized that if you cannot smell anything, then most likely you cannot taste anything either. So that is how they are interlinked at some point. And they work almost the same way. The receptors for taste are also chemoreceptors, but these ones are designed to detect the presence of chemicals in the food that we eat. And so they are located in the mouth. We have receptors found in the tongue mainly, but we also have some on the sides of the mouth. The slide showing some of the structures that are found in the tongue, shaped in different ways, and we have different types of papillae there: filiform, valate, fungiform, foliate, and taste buds. The taste buds, of course, are the structures where the cells that will perceive presence of chemicals in the food that we eat and convert them to action potential. That's where they are located. So check out this in your book, so you are able to identify each of those different types of structures on the surface of the tongue. And again, this is this epithelium containing the taste receptors is replaced every about 10 days. There is a very high rate of replacement of this epithelium. And I'll ask you to check out these pictures in your book. These are the tongue, and you have different structures that we just listed previously, the different types of papillae. And the insert there that is in black and white is a scanning electron micrograph of the surface of the tongue, showing you how it looks like. It's a very complex, very highly organized structure. And inside there is where you find the taste receptors. And the figure on the right shows you the epithelium lining the tongue, where you actually find the taste buds. And the taste buds are modified neurons, and you can see how they look like there on the right side. At very high power, you can see the single cell. The right bottom is where you find a single cell that perceives chemicals in the food we eat and transfers that information to a neuron that takes the information to the brain for interpretation as taste. So check out all these figures and be able to identify those different organs, so that if I give this to you in an exam scenario, for example, you should be able to identify those parts. Now, in contrast to smell, where we can detect up to 4,000 different smells, the number of receptors for taste are limited, so that as of now, we are only able to identify 5 different types of tastes. And each of these tastes are identified by different receptors. So we have 5 different types of receptors. And the tastes that we are able to identify are as follows: sour, salty, bitter, sweet, and umami. These are triggered by presence of different chemicals in the food. So sour taste is triggered by the presence of acid in the food we eat in. So if you are drinking milk that has become sour, for example, there is some kind of lactic acid in that milk, and so it's going to trigger these receptors. Anything that has acid, if you eat oranges, that one has some acid in it. Oranges, most of the fruits actually have some amount of acid in it. Those are the ones that trigger these receptors, and we interpret